Something is already happening at the surface of your skin. It has been happening since before you settled into this position, before you closed your eyes, before you chose this particular hour to let the day finish. Your skin is receiving. It receives without pause, without preference, without rest. Pressure from fabric, slight shift in air temperature near your arm, the weight of your own body distributed across whatever surface holds you now. All of it arriving, all of it converting into electrical signal, all of it beginning its journey inward through fibers too small to imagine and too numerous to count. Most of it will not reach you. Welcome to the Sleepy Biologist. Wherever you are listening from tonight, whatever quiet corner or familiar bed or darkened room, something in your nervous system is already doing the work this whole video will slowly describe. You don't have to do anything except let it continue. There are people who study what the body chooses not to feel. Not researchers of numbness, not specialists in damage. Researchers of ordinary healthy filtering. The kind that happens in every body every moment with remarkable precision and no effort at all. What they find again and again is that sensation is not a recording. It is a selection. Your skin right now contains several different populations of sensory receptors. Some respond to pressure, some to temperature, some to the fine mechanical distortions caused by something as light as a breath of air crossing exposed skin. They are each tuned molecularly, structurally to a particular kind of physical change. When that change occurs, ion channels open. Charged particles shift across the cell membrane. A signal begins. That signal travels. It enters the peripheral nerve fiber, moves toward the spinal cord, and arrives at the dorsal horn, a region of grey matter running the length of the spine where sensory input first meets the central nervous system. Here, the signal encounters something it did not encounter before. Other signals. Competition, context, a nervous system that is already busy, already waiting information, already making quiet decisions about what matters. You've probably felt this before, the way a familiar weight disappears, the way clothing stops registering. You put something on and within minutes the sensation is simply gone, not because the fibers stopped responding, but because somewhere in the processing chain the information was reassigned. Filed, deprioritized. The word neuroscientists use for this is habituation at the level of the synapse, but that language doesn't quite capture what it actually is, which is closer to your nervous system deciding that something constant and harmless does not need to keep interrupting. Synaptic transmission between sensory neurons and the interneurons they contact in the spinal cord is not fixed. It is plastic in the short term, meaning the strength of the connection changes based on recent activity. When the same signal arrives repeatedly in a short window, the presynaptic neuron releases slightly less neurotransmitter with each successive firing. The receiving cell responds less completely. The signal that exits the spinal cord toward the brain carries less amplitude than the one that entered from the periphery. This is happening right now. In the skin touching this surface, in the pressure points along your back or side or wherever weight has been resting for several minutes. The signal that left those receptors was real. The electrical event was genuine. And yet by the time it had travelled a few centimetres into your spinal cord, it had already begun to lose importance. There is a structure in the brainstem called the reticular formation. It is not a structure most people have any reason to know exists. It sits deep, embedded in the core of the brainstem, and among its many functions is something called sensory gating, the process by which certain signals are permitted to continue toward the cortex and others are quietly redirected or suppressed before they arrive. It is not deciding based on conscious preference. It has no access to what you want to feel or don't want to feel tonight. It operates on older criteria: novelty, intensity, relevance to survival, and the current state of the brain it is serving. A brain engaged in focused attention presents differently to the reticular formation than a brain in its current state. The one behind your eyes right now beginning to soften at the edges. The gating shifts. The threshold for what gets through rises slightly. Not all signals, but most routine ones, begin to require more intensity before they earn passage. Your skin is still reporting, the reticular formation is already less interested. There is something worth sitting with, even briefly. The signals that don't make it through are not erased. They are not destroyed at the synapse or dissolved somewhere in the spinal cord. In a biological sense, they simply stop mattering. The cascade that would have led to conscious perception never completes. Nothing accumulates, nothing registers, nothing becomes experience. They existed. They were electrochemical events. As real as anything the nervous system produces, they carried physical information about the world in contact with your body. And then they were over. You have no record of them. There was never a moment when they were close to becoming something you could notice. The threshold was simply never reached, and the absence of their arrival produced no gap, no silence, no awareness of missing information. The body does this with extraordinary volume. Researchers studying single nerve fibers in anesthetized animals have recorded continuous low-level firing from mechanoreceptors under conditions that, in a waking human, produce no sensation whatsoever. The neurons fire, the signal begins its path. Somewhere short of experience it ends. This is not a flaw. It is, in the technical sense, exactly what is supposed to happen. Your body right now contains something on the order of 1 million sensory nerve fibers in the skin alone. Not all are active simultaneously, not all are firing at the same rate. Not all are in contact with stimuli that meet their individual thresholds, but many are. Many are doing exactly what they were built to do, converting physical contact into electrical language and sending that language inward. The cortex, if it received all of it, would not be able to process it. Not in a figurative sense, literally, in terms of available processing capacity and the metabolic cost of that processing. The brain runs on a fixed and limited energy budget, roughly 20% of the body's total energy consumption, in a structure that accounts for roughly 2% of body weight. Every signal that reaches cortical processing costs something. The signals that never arrive are, in a precise sense, the body conserving itself. What does reach you tonight, right now? The pressure that has shifted since you last moved. The slight temperature change at an exposed area of skin. Perhaps something faint and specific, the texture of fabric at a single point, or a subtle awareness of your own breath moving. These signals earned their way through. They were different enough, recent enough, or just barely above threshold in a system that is slowly raising the bar. The rest is arriving continuously. Converted, transmitted, entering the spinal cord, meeting the first synaptic junction, and there, quietly losing the competition for the brain's attention. Not blocked, not damaged, just outweighed. There is a kind of precision in this that is difficult to hold in mind for long, not because it is complicated, but because it describes a process so continuous and so automatic that the mind tends to slide past it. The filtering does not announce itself. It produces no sensation, generates no detectable absence. It simply happens, beneath the level of anything you will ever directly know about it, while you rest in the experience of a body that feels remarkably quiet. The signals are still leaving the skin. They are travelling the same paths they always travel, arriving at the same junctions, meeting the same decisions. But the brain receiving their diminished echoes is already a little slower than it was an hour ago, a little less interested in novelty, a little more willing to let the constant things remain constant. What gets through will keep getting quieter. Not because the body is going, "What? It won't, not entirely, not tonight," but because the systems that decide what matters are beginning to apply different criteria. What counts as important starts to narrow. The field of incoming sensation begins, very slowly, to lose its edges. Somewhere in the next layer of this, there are signals that made it further. Ones that entered the thalamus, moved toward the cortex, almost became perception. What happened to them, and why some things crossed that final distance while others stopped just short of it, is a question the body has been answering long before there was any language to ask it. There is a number that gets cited in neuroscience courses, passed between researchers, repeated in textbooks until it starts to feel less like a measurement and more like a claim. The estimate: the skin contains somewhere between 1 and 5 million individual sensory receptor endings, depending on the method of counting, the region of the body, the species being studied, the assumptions built into the model. It's a large number. But what stands out when you stay with it is not the number itself. It's what the number implies about the scale of what is currently being discarded. Right now, the surface of your body is in contact with more than one thing simultaneously. Fabric has its own texture, not smooth under sufficient magnification, but a structure of interlocking fibers, each pressing against skin at slightly different angles, each engaging a slightly different subset of mechanoreceptors. The air above exposed skin carries temperature. Beneath you, pressure distributes across a surface that is never perfectly uniform, creating gradients of compression that shift whenever you breathe. All of this is being transduced, converted. The receptors involved are not generic. Meissner's corpuscles sit near the surface of the skin in ridged areas, fingertips, lips, the soles of feet, and respond to light touch and motion across the skin. Merkel discs, slightly deeper, register sustained pressure and fine spatial detail. Pacinian corpuscles, deeper still, are tuned almost exclusively to vibration and rapid changes in pressure. They are among the fastest adapting receptors in the body, meaning they fire briefly at the onset of a stimulus and then, within milliseconds, fall quiet even if the stimulus remains. The stimulus remains. The receptor goes quiet. This is not a failure of function. It is the function. Each receptor type speaks a slightly different dialect of the same electrical language. The signal that leaves a Pacinian corpuscle carries different temporal information than the one leaving a Merkel disc under sustained pressure. They travel in different fiber types, some fast, some slow, some carrying only the broad outlines of what happened, some transmitting fine detail with higher fidelity. They converge in the dorsal horn of the spinal cord, where the conversation becomes more complicated. At this junction, sensory fibers do not simply pass their signals upward and continue. They synapse. They release neurotransmitters onto interneurons, cells that do not themselves extend to the skin or to the brain, but that mediate, modify, and in some cases, suppress what happens next. Some of these interneurons are inhibitory. Their activation does not amplify the incoming signal, it quiets it. The signal arrived. An inhibitory cell responded. What continues upward is already different from what came in. It's not entirely clear, even now, exactly which signals get selected for suppression at this level versus at higher stations in the brain. The criteria aren't fully mapped. There are models involving signal intensity, repetition rate, competing inputs from other fibers, descending instructions from the brainstem, but the whole picture remains partially unresolved. What is clear is that the selection is not random. Certain features reliably survive the first filter. Rapid change in stimulus intensity, stimuli arriving at locations not recently activated, inputs that arrive during a window of low competing activity. The nervous system appears to have a preference, if that word can be used, for information that is new relative to what it was just receiving. What was already there tends to disappear. Your skin has been in contact with its current environment for some time now. The fabric has not moved. The pressure beneath you has not shifted significantly. The temperature at the surface has reached something close to equilibrium, your body warming the immediate air around it until the gradient between skin and environment is barely a gradient at all. The receptors sensitive to all of this are still there. Some are still firing, but they are firing into a system that has already evaluated this configuration and, through mechanisms that require no deliberation and produce no sensation, determined that it does not require further reporting. You're not feeling the full surface of your body right now, you're feeling its exceptions. There is a category of receptor that responds specifically to the boundary between two different skin temperatures, not to heat itself, not to cold itself, but to the precise location where one condition transitions to another. These receptors exist, not to tell the nervous system what temperature the body is at, but where a gradient begins. The edge, the difference, the departure from what was already known. This is a principle running through sensory biology at nearly every level. The system is not built to report states. It is built to report changes. A steady input, no matter how present, no matter how continuous, tends to lose its claim on the circuits built to respond to it. What remains constant begins to disappear. What changes returns. You've probably felt this in the opposite direction, the way re-entering a familiar scent makes it briefly vivid again after it had vanished from awareness. Or the way shifting position in the dark makes you suddenly aware of the weight of fabric that had been moments before undetectable. The change re-engaged what the constancy had allowed to go quiet. The signal hadn't stopped, the reporting had. This quality of sensory system, their bias toward novelty, their partial blindness to what is sustained, is sometimes framed as a limitation. It's possible it is more accurately described as a design system that continuously reported the full state of all surfaces simultaneously would be generating an enormous and largely redundant stream of information. Most of what the skin is in contact with most of the time is the same as what it was in contact with a moment ago. The body has already accounted for that. The architecture of forgetting in sensory systems is older than the nervous systems we recognize today. Simple organisms without a brain in any conventional sense still exhibit habituation, a reduction in response to repeated stimulation that cannot be explained by fatigue in the receptor itself, because the response recovers when the stimulus is changed. The selectivity predates complexity. It may be among the earliest solutions to the problem of having a body that interacts with a world that is partly stable and partly unpredictable. What keeps changing, pay attention to. What stays the same, release. Right now, the surface of your body is in the second category. The fabric is the same. The pressure is the same. The temperature is close enough to what it was a few minutes ago that the receptors measuring it have little new to say. What the skin is keeping to itself tonight is the ordinary record of ordinary contact, the texture of what holds you, the weight of what covers you, the temperature of a surface that has had time to meet yours. It filed all of it, and then, quietly, it stopped sending. What remains in awareness is less than a summary. It is the residue left after the residue was cleared. The next level of this filtering does not happen at the skin, or even at the first junction in the spinal cord. It happens deeper, in structures that receive input from the spinal cord only after it has already been reduced once, and that apply their own criteria to what continues. What those criteria are, and what they allow through, belongs to what comes next. In 1965, two researchers published a paper that changed, in a fairly fundamental way, how the nervous system's handling of pain and touch was understood, and the central idea they propose was elegant in the way that certain biological explanations are elegant, not because they simplify something complicated, but because they make visible a structure that was always there, operating without anyone having named it yet. Ronald Meltzack and Patrick Wall described what they called a gate control mechanism in the dorsal horn of the spinal cord, a place where, they proposed, the transmission of pain signals could be modulated by the simultaneous activity of other sensory fibers, specifically the large diameter fibers that carry touch and pressure, and the implication of this, quietly radical for its time, was that the spinal cord was not a passive relay, not a simple wire carrying signals upward to a brain that would then decide what to do with them, but an active site of processing, a place where the relationship between what arrived from the periphery and what continued toward the brain was being continuously negotiated. The gate, as they described it, could be opened or closed. Touch, in their model, could close it, could reduce the transmission of pain signals by activating inhibitory interneurons in the dorsal horn that dampen the relay cells responsible for sending pain information upward. And this is why rubbing a bruised area produces something that is not exactly pain relief but is recognizably different from letting the area sit untouched. Why the instinct to press a hand against something that hurts has a real, if partial, physiological basis. Why the nervous system appears to have built into itself a mechanism by which one kind of sensation can, under certain conditions, reduce the passage of another. It was a useful model. It directed decades of research and remains part of how pain medicine thinks about central sensitization, about spinal cord stimulation as a therapeutic approach, about why certain chronic pain conditions involve changes not just in peripheral tissue, but in the spinal cord circuits that were supposed to be doing the gating. But the gate was never really a gate. The metaphor, which Meltzack and Wall introduced carefully and which subsequent popularization made considerably less careful, implies a binary, open or closed, passing or blocking, and the actual biology resists this, because the dorsal horn is not a structure with two states, but a dense, continuously active network of excitatory and inhibitory interneurons, projection neurons, descending fibers arriving from the brainstem and cortex, propriospinal connections running between spinal segments, and the interaction between all of these produces something closer to a continuously shifting probability, the likelihood at any given moment that a given incoming signal will cross the synaptic distance to the projection neuron that will carry it toward the brain. That probability changes. It changes based on the recent history of the synapse, based on what other fibers are active nearby, based on the concentration of neuromodulators released by descending systems, based on the state of the inhibitory interneurons themselves, which have their own thresholds and their own recent histories, and understanding all of the conditions that move that probability up or down in any particular person at any particular moment remains genuinely incomplete. The gate was a way of seeing something real. The something real was more complicated than any gate. What the model captured, imperfectly but usefully, was the principle that the spinal cord participates in a conversation rather than a transmission, that the signal arriving from the skin is not simply forwarded, but evaluated in the context of everything else, arriving at the same time, and this remains true even after the metaphor has been softened, and the specific circuit diagram has been revised, and the researchers who came after Meltzack and Wall have mapped the interneuron populations with considerably more precision than was available in 1965. The dorsal horn contains, among others, a population of inhibitory interneurons that use GABA and glycine as their primary neurotransmitters, and when these cells are active, they reduce the excitability of the projection neurons that would otherwise carry nociceptive signals toward the thalamus, and eventually toward the cortex, and when these inhibitory cells are themselves suppressed by injury, by inflammation, by certain descending inputs, the projection neurons become more responsive, requiring less input to fire, broadening the range of stimuli that reach conscious processing. This is part of what happens in central sensitization. The inhibitory interneurons become less active. The gate, to use the language that is no longer quite accurate but remains evocative, swings open in a direction it was not meant to sustain. But this is not where you are tonight. Tonight, the inhibitory interneurons in your dorsal horn are receiving descending input from the brainstem, from the periaqueductal grey matter and the rostral ventromedial medulla, structures that release serotonin and norepinephrine into the spinal cord in a way that tends to enhance inhibition, that tends to close whatever the gate actually is, that tends to reduce the upward transmission of signals that a more aroused nervous system might have allowed through without question. This descending modulation is not a deliberate act. You're not choosing it. It is part of a coordinated shift in the state of the nervous system that happens as wakefulness recedes, as the brainstem changes its output, as the whole system moves slowly and in parallel toward a different configuration. The signals arriving at your dorsal horn right now are meeting a different set of conditions than they would have met this afternoon, and a different set still from what they will meet an hour from now. There is something worth holding gently here, not as an argument but as an observation, the recognition that the processing of sensation is not a fixed property of the nervous system but a state-dependent one, that the same stimulus arriving at the same receptor can produce different outcomes at the level of the spinal cord depending on what the rest of the nervous system is doing at the time, and that this is not an exception or an anomaly, but the ordinary, continuous operation of a system that was never designed to process sensation in isolation from everything else happening in the body at the same moment. The gate was never a gate because the structure it described was never binary, never static, never independent of the organism's current condition. It was always a relationship between arriving signals and waiting circuits, between the periphery and the center, between what the body just experienced and what it is about to decide to feel. And right now, in the quiet of wherever you are, the arriving signals are meeting circuits that are already in the process of becoming less interested. And the relationship between the two is shifting in the direction it shifts every night in every body that is moving towards sleep. The inhibitory tone increasing, the threshold rising, the probability that any given peripheral signal will successfully complete its journey to conscious experience declining gradually without announcement, without a moment that could be identified as the moment it changed. The dorsal horn does not know that you are trying to sleep. It knows only the neurochemical conditions it is currently receiving, and those conditions are telling it, in the language of descending modulation and inhibitory tone, to quiet the upward traffic. The signals are still arriving at the gate that was never a gate. Fewer of them are getting through, and the ones that do not get through leave no trace of having tried, no echo, no residue, no awareness of an absence where a sensation might have been. They arrived, and the conditions were not right, and they dissolved back into the silence that surrounds all the signals the body decides, in its quiet and continuous way, not to feel. You already know this, in a way, not from reading about it, from having a body, from having been somewhere loud and then, after a while, not hearing the loudness anymore, from having worn something uncomfortable that, an hour later, you forgot you were wearing, from having had a headache that disappeared, not because it ended, but because something else took over, and the headache somehow moved to the background. You've noticed this, maybe not as a phenomenon worth examining, just as a thing that happens. It turns out the thing that happens has a physical basis that is, and this is the part that takes a moment to sit with, not located where you might expect. The filtering of sensation based on attention doesn't happen only at the skin, or only in the spinal cord, or only in some dedicated suppression system. It happens in the cortex itself. In the very place where sensation becomes experienced, there are circuits whose primary function is to reduce their own responsiveness to certain inputs, to actively dampen the processing of signals that have been deemed, by some prior layer of the system, not worth full attention right now. Active. That's the word that keeps coming up in the research. The brain doesn't passively fail to notice things. It works, uses energy, recruits circuits, issues instructions downward through the very pathways that sensory signals travel upward to not notice them. There's a structure called the pulvinar, part of the thalamus sitting toward its posterior end, and one of its roles involves something researchers describe as attentional gating, the selective amplification of some sensory streams and the concurrent suppression of others based on where attention is currently directed. When attention moves, the pulvinar adjusts, not slowly. Within fractions of a second, the gain on certain sensory channels changes. What was being amplified quiets, what was being suppressed gets a little louder. The signal from your skin didn't change, the attention did, and the attention changed what the signal was worth. This is, and it takes a moment to find the right way to say this, a resource problem, not a limitation exactly, more like an economy. The cortex cannot process all incoming sensory information with equal depth simultaneously, not because the neurons aren't there, but because deep processing requires sustained coordinated activity across multiple areas, requires the allocation of something that functions loosely like processing bandwidth. And that bandwidth is finite in a way that has real consequences. The visual cortex receiving input from both visual fields does not process both fields with equal resolution. It processes the attended region more completely. The unattended region is processed, but shallowly, enough to flag sudden changes, enough to detect motion, not enough to build a full detailed representation of what is there. The same principle operates across sensory modalities. What is attended to is processed deeply. What is not attended to is processed just enough. Just enough for what, though? This is where it gets not complicated exactly, but interesting in a way that doesn't resolve into a clean answer. The shallow processing of unattended stimuli is not nothing. It's a kind of standing watch. The brain is not ignoring the periphery, it's monitoring it at low cost, ready to redirect attention if something there changes enough to warrant it. A sudden sound, a sharp pressure, a temperature shift large enough to suggest something has changed in the environment. These can break through, not because the attentional gate failed, but because the shallow processing detected something that met the threshold for escalation, for pulling attention away from wherever it was and redirecting it toward the new signal. The system is not a lock. It's a priority queue, and the priority queue is, right now, tonight, running differently than it was this afternoon. When wakefulness is high and the environment is demanding, the queue is long and the thresholds for escalation are relatively low. Many things get flagged. Many things get pulled into fuller processing. The sensory environment feels more present, more detailed, more insistent. As the brain moves towards sleep, the queue shortens, the thresholds rise. What would have escalated an hour ago sits in the shallow processing layer and does not move. This is partly driven by the thalamus, which acts as a kind of relay station between the periphery and the cortex, and which during the transition to sleep begins generating rhythmic activity, spindle activity eventually, that partially closes the sensory relay. The pulvinar shifts its gating. The cortex becomes less responsive to thalamic input, even when thalamic input is still arriving. Attention, which was always a biological resource, begins to be allocated differently. less of it outward, less of it to the surface of the body, to the sounds in the room, to the low-level environmental signals that were being monitored without your knowing they were being monitored. Where does it go? Inward, partly. There is evidence that the transition to sleep involves a shift in the direction of the default mode network, the set of cortical regions active during the internal thought, self-referential processing, the kind of thinking that doesn't require engagement with external input. This network becomes more active as external attention decreases, which is one reason the period before sleep is often populated by drifting thoughts, half-formed images, memory fragments that arrive without invitation. The attention hasn't disappeared, it's been redirected, away from the skin, away from the room, away from the sounds and pressures and temperatures that the sensory system has been quietly monitoring, and the attentional system has been quietly suppressing or elevating based on criteria you never had access to. The fabric you're lying on, it's still there. The receptors are still doing what receptors do. The signal is still being generated, still travelling, still arriving at the thalamus. But the pulvinar is less interested. The cortex is less responsive. The resource that would have been used to bring that signal into full experience is being used for something else, or not being fully used at all. The sensation doesn't disappear, it just stops being worth the cost. And there is something almost settling about that, not as a metaphor, but as a literal description of what is happening in the tissue and the circuits and the allocation systems right now, as whatever was holding your attention to the edges of your body begins, quietly and without decision, to let them go. The next layer of this is not about attention at all. It's about what happens when the signal does reach the cortex, when it makes it through every prior filter, arrives at the place where experience is assembled, and still somehow fails to become something felt. The thalamus is not a single thing. This is worth stating plainly because it tends to be introduced as one, a relay station, a sensory hub, a central switchboard, and while none of those descriptions are wrong, they flatten something that is considerably more differentiated than any single label suggests. The human thalamus contains somewhere between 50 and 60 distinct nuclei, depending on the classification system used. Each has its own inputs, its own targets in the cortex, its own role in the larger economy of sensory processing. Some relay visual information, some handle auditory signals, some are involved in motor coordination, in attention regulation, in the modulation of cortical arousal, and some, particularly the ventral posterior nucleus and, as discussed briefly before, the pulvinar, are centrally involved in what happens to the sensory signal between the spinal cord and the cortex. What happens there is the part that stands out most when you look at the full architecture of filtering. By the time a signal from the skin reaches the thalamus, it has already been reduced once. The inhibitory interneurons of the dorsal horn have already done their work. The signal arriving at the ventral posterior nucleus is not the raw output of the peripheral receptor. It is a modified version, already shaped by what the spinal cord found relevant. The thalamus receives this and does something that took researchers a long time to fully appreciate. It doesn't simply pass the signal to the cortex, it evaluates the current state of the cortex first. The thalamocortical relationship is bidirectional in a way that is easy to understate. For every fiber running from the thalamus to the cortex, there are roughly 10 running in the opposite direction, from the cortex back to the thalamus. The cortex is not a passive recipient of thalamic output. It is continuously shaping what the thalamus sends it. This feedback loop means that what reaches conscious processing is partly determined by what the cortex is already doing. A cortex engaged in focused cognitive work sends descending signals that modulate thalamic relay cells in ways that can either enhance or suppress the transmission of sensory input from below. A cortex in its current state, quieter, less engaged with external demands, beginning the slow withdrawal from active processing, sends different signals. The thalamic relay cells respond accordingly. What gets through changes. There is a specific mechanism worth pausing on. Thalamic relay cells can operate in two distinct modes, depending on their current membrane potential. In what researchers call tonic mode, they faithfully transmit incoming signals to the cortex with reasonable fidelity. A signal arrives, the relay cell fires. The cortex receives something approximately proportional to what came in. In burst mode, the cells behave differently. When hyperpolarized, held at a more negative resting membrane potential by sustained inhibitory input, they become capable of generating calcium-dependent bursts of action potentials that do not accurately reflect the sensory input that triggered them. The burst is more about the internal state of the cell than about what arrived from the periphery. The transition from tonic to burst mode happens during drowsiness and early sleep. This is the part that feels most significant when you trace it fully. The relay itself changes character. The thalamus stops relaying and starts generating. The signal that reaches the cortex during this transition is not a faithful representation of what the skin reported. It is a distorted version, shaped by the state of the cells doing the transmitting. What the cortex receives then is already a version of a version of a version. The receptor fired, the spinal cord modified the signal, the thalamic relay cell, now trending toward burst mode, modified it again. What arrives at the primary somatosensory cortex, the region of the cortex that handles the initial processing of touch and body sensation, is something that has passed through three distinct layers of transformation. Each layer applied its own criteria. None of the criteria were yours. The primary somatosensory cortex is organized somatotopically, meaning different regions of the body are mapped onto different regions of the cortex in a way that is spatially organized if not proportional to actual body size. The hands and face occupy far more cortical territory than the back or the thighs, reflecting the density of sensory receptors in those areas and the precision of discrimination required there. This map is not fixed. It is plastic over time, meaning the territory allocated to different body regions can expand or contract based on use, on injury, on sustained patterns of sensory input or its absence. But its plasticity is slow. The map you have tonight is largely the map you had last year. It was built over decades of sensory experience, each pattern of input leaving small changes in synaptic strength, in dendritic branching, in the receptive fields of cortical neurons. The filtering that happens at the cortex operates on top of this map. What reaches S1, the primary somatosensory area, still has to compete for processing within a cortex that is beginning to disengage, within circuits whose threshold for sustained response is rising, within a region that will, within the next layer of sleep, become partially isolated from the thalamic input it normally depends on. There is no single location where the decision is made. This is the part that resists clean description. Researchers have traced the sensory pathway from receptor to cortex with considerable precision, have identified the inhibitory interneurons, the thalamic relay modes, the corticothalamic feedback, the modulating influence of brainstem nuclei on the whole system. And yet there is no structure that can be pointed to as the place where the filtering happens, no nucleus whose removal would result in all sensations getting through, no circuit whose activation would suppress them all. The filtering is the system. It is the aggregate output of a network operating in a particular state, and the state changes, and the output changes with it, and what you feel at any moment is what survived all of it. Right now the state is shifting, the thalamic relay cells are beginning to hyperpolarize, the corticothalamic feedback is quieting. The primary somatosensory cortex is receiving less input than it was an hour ago, and processing what it receives less completely. The signals from your skin are still arriving at the thalamus. Some are still making it through to the cortex, but the cortex that receives them is not the same cortex that would have received them this afternoon. It is a cortex in the early stages of letting the sensory world become less important than whatever is happening inside it. The filtering hasn't stopped. It has simply moved to a deeper layer. And what it is filtering now is not just sensation, but the relevance of sensation itself. The question of whether any of this, the fabric, the pressure, the temperature, the whole continuous report of a body in contact with its environment needs to be attended to at all. It doesn't. And somewhere in the circuits that just arrived at that conclusion, something begins to settle that has nothing to do with the signal still travelling upward, still arriving, still waiting for a cortex that is already quietly looking elsewhere. You have stopped noticing the weight of your head. At some point tonight, maybe when you first settled, maybe a few minutes after, the pressure of your head against whatever supports it registered, briefly and then receded. Not because the pressure changed, because you stopped being asked about it. This happens with almost everything the body holds for long enough. There is a name for the cellular mechanism behind this: synaptic depression. It describes what happens at the junction between two neurons when the same signal arrives repeatedly in a short window of time, when the presynaptic neuron is asked again and again to release neurotransmitter in response to the same incoming pattern. The first release is complete. The vesicles, small membrane-bound packages of neurotransmitter clustered near the synaptic terminal, fuse with the membrane and empty their contents into the synaptic cleft. The postsynaptic cell receives the chemical signal and responds, but the vesicles take time to replenish. The second signal arrives before replenishment is complete. Fewer vesicles are available. Less neurotransmitter is released. The postsynaptic response is smaller. The third signal, smaller still. This is not damage. It is not exhaustion in any harmful sense. It is a property of the synapse itself, a built-in responsiveness to the history of its own activity. The synapse remembers, in a purely chemical sense, what it was just asked to do, and it adjusts. You know this feeling from outside the nervous system too. The first sip of something is the most vivid. The first few seconds of a hot shower are the most present. The first minute of cold water is the most insistent. After that, the signal is still arriving, but the response to it has already shortened. The stimulus did not change, the synapse did. Synaptic depression operates across different timescales. Short-term depression, the kind involving vesicle depletion, resolves within seconds to minutes once the stimulus stops or slows, the terminal restocks. The next signal, if it arrives after a sufficient pause, meets a fully prepared synapse and produces a full response. This is why a break in sensation can restore it, why stepping back into cold water after a moment out of it makes it cold again, why the weight of your head would, if you moved and resettled, briefly return to awareness before disappearing once more. The pause resets the synapse. The synapse resets the signal's power to reach you. But there is a deeper layer of adaptation that operates over longer timescales and involves not just the synapse, but the receiving neuron itself. When a postsynaptic neuron is repeatedly activated by the same input pattern, it can reduce its own responsiveness through a process involving receptor internalization, the physical withdrawal of neurotransmitter receptors from the cell surface into the interior of the cell. Fewer receptors on the surface means less response to the same amount of neurotransmitter. The neuron has, in a real molecular sense, made itself less available to this particular signal. This process, depending on the receptor type and the duration of stimulation, can take minutes to hours to reverse. It is a slower, more committed form of adaptation. The synapse that has undergone receptor internalization is not simply depleted. It has structurally adjusted to a world in which this signal is routine. What is routine does not need a full response. Think of a sound that was once startling, an alarm, a note of satin, a particular tone, that over time became simply present, then barely present, then essentially absent from awareness despite still occurring at the same volume. The ear did not change. The auditory nerve continued to transmit. But somewhere in the chain of synapses between the cochlea and the cortex, the response shortened, the receptor density adjusted, the neural representation of that sound became thinner, less complete, less capable of reaching the circuits that produce conscious awareness. The nervous system had filed it, not deleted, filed. Still there if queried, still capable of producing a full response if the sound changed significantly, still represented in the sensory system, but no longer generating the kind of sustained cortical activity that experience requires. Right now, this process is running across most of the sensory surface of your body. The receptors in your skin have been reporting the same contact for long enough that the synapses carrying their signals have been through multiple cycles of activation and partial depletion. The signals are arriving with less downstream consequence than they had 20 minutes ago. The neurons receiving them in the spinal cord are responding less completely. Some have begun the slower process of receptor adjustment. This is accumulating quietly, without any single moment you could identify as the moment it happened. The weight of your body against the surface holding it is being represented in your nervous system with less and less resolution, not as an act of forgetting, but as the natural output of a system that has evaluated this configuration, found it stable, found it non-threatening, found it continuous, and begun the cellular process of treating it as background. There is something almost careful about the way the nervous system handles what is familiar, not careless, careful. It does not abandon the monitoring of surfaces in contact with the body. It maintains a standing watch, as mentioned before, ready to restore full responsiveness if something changes. But it holds that watch at minimum cost, with the smallest activation necessary to maintain readiness. The synapses are quiet but not closed. The receptors are reduced but not absent. The signal is thin but still travelling. What has changed is only the weight it carries when it arrives. Your nervous system has been in contact with this environment for long enough, now that almost nothing about it is surprising. The temperature has stabilized, the pressure distribution has been logged. The texture of whatever is in contact with your skin has been sampled and released. The synapses carrying all of this have had time to adjust. What arrives at the thalamus now is already the diminished output of a system that has seen this before, recently, repeatedly, and reduced its response accordingly. What the thalamus passes to the cortex is reduced again. What the cortex receives is a thin signal representing a body at rest in a familiar environment, and the cortex, already in the early stages of its own withdrawal from sensory engagement, accepts this thin signal and does very little with it. You are still receiving sensation. The chain is still intact. It has simply become very quiet along its entire length. The next question, and it is one the nervous system has been working on since long before there was language to ask it, is what happens when a signal that has been reduced this far, thinned across 3 or 4 synaptic layers, still manages to arrive at a cortex that is nearly done listening, whether it produces anything, whether it reaches whatever threshold separates a signal from an experience, and what exactly that threshold is made of. There is a flatworm, planaria, that has no brain in any sense that maps comfortably onto the word, no cortex, no thalamus, no dorsal horn, no descending modulatory pathways, no pulvinar nucleus performing attentional gating on competing sensory streams, and yet when you expose it to the same mild stimulus repeatedly, its response diminishes, not because its body has been damaged, not because the stimulus has changed, but because something in the distributed network of neurons running through its body has adjusted to the pattern of arrival, has registered the repetition, has, in whatever way is available to a creature without a center, decided that this particular input no longer requires the same degree of response. It habituates. This observation, which has been reproduced across dozens of invertebrate species, and which holds in organisms with nervous systems far simpler than anything that could be called a brain, carries a weight that is easy to move past too quickly, because what it suggests is that the logic of sensory filtering, the principle that repetition reduces response, that novelty earns attention, that what is sustained loses its claim on the circuits built to process it, is not a feature of complex nervous systems that evolved alongside consciousness, but something considerably older, something that was solved once, very early, and then carried forward more or less intact through every subsequent elaboration of nervous tissue. The solution predates the problem as we currently understand it. The cellular mechanism underlying habituation in simple organisms involves changes at the synapse, specifically a reduction in the amount of neurotransmitter released by the presynaptic neuron in response to repeated activation, along with a reduction in the number of active synaptic connections between the sensory and motor neurons involved in the habituated response. Changes that Eric Kandel and his colleagues mapped with extraordinary precision in the sea slug Aplysia californica during the 1960s and 1970s, work that eventually produced a Nobel Prize, and that revealed, among other things, that the synapse itself, not just the network, not just the circuit, but the individual junction between two neurons, was capable of storing information about its own recent history in a way that changed its future behavior. The synapse remembers what it was just asked to do. This is old, this is very old. What Kandel found in Aplysia was that short-term habituation involved functional changes at existing synapses, the depletion and incomplete replenishment of synaptic vesicles, the reduced probability of neurotransmitter release, while long-term habituation involved something more structural, a reduction in the actual number of synaptic connections between sensory and motor neurons, a physical retraction of the synapse itself that required protein synthesis and that persisted for days or weeks after the original training. The memory of the repeated stimulus was written into the structure of the nervous tissue, not metaphorically, physically. The neurons that had been repeatedly activated together had fewer connections afterward than they had before. I tend to think this is the part of sensory biology that sits most quietly in the background of everything else. The recognition that the filtering happening in your nervous system right now is not happening in some specialized, recently evolved structure dedicated to the sophisticated management of sensory experience, but in the same basic mechanism that a sea slug uses to stop withdrawing its gill when the same water current arrives for the 10th time in a row, a mechanism so fundamental that it has been conserved in its essential logic across 600 million years of evolutionary distance. Your spinal cord interneurons reducing their response to the fabric against your skin tonight are doing something that is, at the level of the synapse, recognizably continuous with what Aplysia's sensory neurons do when they have been stimulated one too many times. The elaboration is different. The principle is the same. What evolution did over the time between Aplysia and the nervous system you're currently using was not replace this mechanism but build around it, adding layers of complexity above and below, adding descending modulation and thalamic gating and cortical feedback and attentional systems that could actively direct the filtering rather than simply allowing it to emerge from synaptic depletion, but never removing the original solution, never finding a reason to retire, the basic logic that repetition should reduce response and novelty should restore it, because that logic remains correct regardless of how complex the organism processing it becomes. The ancient solution still works. There is something in this worth resting with for a moment, not as a scientific argument, but as a simple observation about continuity, about the relationship between the body you inhabit tonight and the far simpler bodies that solved this same problem long before there was anything like awareness to be protected from sensory overload, about the way the mechanisms running below your consciousness right now carry, embedded in their structure, a history that no individual life could contain. Your synapses are doing something tonight that nervous tissue has been doing, in various forms, for longer than there have been vertebrates. The filtering is ancient, the body performing it is new. The two have met, as they do every night, in the ordinary silence of a nervous system that is, without drama, doing what nervous systems have always done when the environment stops changing and the signals stop bringing news. It reduces its response, it waits. The waiting is not passive in any strict biological sense. The synapses are still capable, the receptors are still present, the circuits are still intact and ready to restore full responsiveness if something arrives that breaks the pattern of familiarity, but it has the character of passivity from inside, from the position of being the organism in which all of this is occurring. Because what the waiting produces is precisely nothing detectable, precisely no sensation, precisely no signal reaching the level where signals become experience. What it produces is silence, and the silence is not empty but structural, built from the accumulated adjustments of 1,000 synapses each doing the same small thing, releasing a little less, responding a little less completely, representing a little less of what the periphery is still faithfully reporting. The flatworm in the water stopped responding to the current. Your nervous system tonight is doing something that rhymes with that, quieter, more layered, more distributed across more structures than a flatworm could contain, but recognizable across the distance as the same old answer to the same old question. The signal keeps arriving, and somewhere in the tissue that receives it, older than memory, older than sleep itself, the decision keeps being made to let it pass unnoticed. It's easy to forget that the nervous system was not designed for the sensory environment most people currently inhabit, not designed for continuous low-level noise from ventilation systems and distant traffic and the faint electromagnetic hum of devices that are never fully off, not designed for the sustained artificial light that keeps the retina reporting long past the point where, for most of human evolutionary history, it would have gone dark. Not designed for the particular quality of modern stillness, which is not really stillness at all, but a background of low-intensity, unvarying, unresolvable signal that never quite rises to the threshold of demanding attention and never quite falls silent enough to stop being registered. The nervous system was designed, shaped, selected, refined for an environment that was quiet when it was quiet and loud when it was loud, that provided genuine contrast, genuine novelty, genuine silence. What it received instead, somewhere in the last century, was continuous mild input with very little dynamic range. The response to this, at the level of the individual nervous system, has been what the response to any sustained input tends to be: a gradual adjustment of baseline sensitivity, a recalibration of what counts as signal and what counts as background, a slow upward drift in the threshold required before incoming information earns a response. You start to notice this most clearly in its absence. The way a truly quiet environment, a forest, a room without electronics, a night genuinely free of distant sound, can feel, initially, almost loud with its own particular quality of nothingness. The auditory system, calibrated to a baseline that includes the usual low-level modern noise, registers genuine quiet as a kind of presence, as something that is different from what it expected. The threshold has shifted. The baseline has risen. What is normal has been redefined at the level of the synapse. Central sensitization is the name given to the pathological end of this spectrum, the condition in which the nervous system's sensitivity becomes amplified rather than reduced, in which the thresholds that normally filter routine sensation downward instead drift upward in the wrong direction, in which stimuli that should produce mild or negligible responses instead produce pain, hyperalgesia, allodynia, the clinical term for when things that are not painful become painful, when light touch produces burning, when the weight of fabric against skin becomes something that cannot be tolerated. This is the filter failing in the direction of too much rather than too little. It involves changes at many of the same locations already described: the dorsal horn into neurons, the thalamic relay cells, the corticothalamic feedback circuits. But in central sensitization, these changes move in the opposite direction from the ordinary adaptive filtering. Instead of inhibitory tone increasing, it decreases. Instead of thresholds rising, they fall. The circuits that would normally dampen incoming signals begin to amplify them, and the amplification, once established, tends to sustain itself through mechanisms involving the long-term potentiation of synaptic connections in the spinal cord, the same basic cellular process that underlies learning, applied here not to memory but to pain. The system learned to be louder. What makes central sensitization particularly relevant to a discussion of ordinary sensory filtering is not the pathology itself, but what its existence reveals about the normal state, about the degree to which ordinary healthy sensory processing depends on active, continuous, resource-consuming suppression of incoming signals, and about what happens when that suppression is interrupted or overwhelmed. The inhibitory interneurons of the dorsal horn require energy. They require ongoing synaptic input from descending modulatory pathways. They require, in a sense, maintenance, not the way a machine requires maintenance, but the way any active biological process requires the continuous provision of the conditions that make it possible. When those conditions are disrupted by sustained inflammation, by tissue damage, by the particular kind of nervous system strain that accumulates in certain chronic pain conditions, the suppression weakens, the threshold falls, and signals that would previously have been intercepted begin to arrive at the cortex with a completeness they were never supposed to have. The background becomes the foreground. It's worth pausing on what this means in the reverse direction, in the direction of ordinary evenings, ordinary fatigue, ordinary nervous systems doing what they normally do at this hour. The descending inhibitory pathways that maintain sensory suppression in the dorsal horn are partly driven by brainstem structures whose output varies with arousal state. When arousal is high, when the organism is alert, engaged, responding to environmental demands, these pathways are active and the inhibitory tone in the dorsal horn is well maintained. When arousal decreases, as it does now, the brainstem output shifts and the descending maintenance of spinal inhibition changes character. This might seem to suggest that sensory filtering should worsen as sleep approaches, that the falling arousal should reduce inhibitory tone and allow more signals through. But the picture is more layered than that. The decrease in arousal-driven descending drive is accompanied by a simultaneous increase in sleep-specific inhibitory mechanisms, the thalamic gating that increases during drowsiness, the cortical withdrawal from sensory engagement, the shift in thalamocortical relay cells toward burst mode, and the net effect in a healthy nervous system making the ordinary transition to sleep is continued or even enhanced filtering just through different mechanisms than those operating during wakefulness. The suppression doesn't fail, it transfers. There is something almost considered about this, the way the nervous system has arranged for sensory filtering to be continuously maintained, across the transition from wakefulness to sleep, even as the specific structures performing the filtering change, even as arousal decreases and certain inhibitory pathways reduce their output, with other mechanisms stepping into the gap so that the transition does not create a window of increased sensory vulnerability. Evolution, in its slow and unsentimental way, seems to have found this transition important enough and protect. A sleeping organism that suddenly became more sensitive to sensory input during the descent into sleep would have been an organism that frequently failed to complete the descent, startled back to wakefulness by the very signals the sleep transition was meant to silence. The arrangement that survives is one in which the handoff between waking suppression and sleep suppression is smooth enough that the ordinary sensory environment does not interrupt it. You're in that handoff right now. The chronic noise that the modern nervous system manages, the low-level acoustic and electromagnetic and photic background of an environment that is never quite dark, never quite silent, has been processed and filed and largely released by this hour. The recalibrated baselines that modern sensory environments tend to produce are not tonight working against you. If anything, the elevated threshold that sustained mild input tends to produce means that the ordinary background of your environment sits even further below the level of conscious detection than it would in a nervous system that had spent its days in genuine quiet. The adjustment that was made in response to chronic low-level noise has raised the floor, and the floor tonight is high enough that very little reaches above it. You start to notice, if you pay attention to the space between your skin and your awareness of it, that the contact is barely there, not absent. The receptors are still reporting, the signals are still travelling, but thin, reduced at every layer, filtered at the spinal cord and again at the thalamus, and again at the cortex that is already withdrawing its attention from the sensory surface of the body. The chronic noise has gone quiet, not because the environment changed, because the system that processes it has reached a state in which almost nothing from outside justifies the energy of a full response. What remains of the sensory world right now is close to the minimum the nervous system can represent while still maintaining its readiness to respond if something genuinely changes. The next question is, what lives just below that minimum? What the body is still doing, still generating, still processing, even in this state of near complete sensory withdrawal, and whether any of it ever becomes something that could be called experience at all. There is a threshold, not a wall, not a boundary in any physical sense, more like a condition that has to be met before a signal becomes something you can call an experience. Below it, the signal exists, it travels, it arrives at structures capable of processing it, and then, without announcement, without any detectable transition from your side of things, it simply does not become anything. It was there, and then it wasn't experience. Researchers studying this threshold have a term for what lives just below it: sub-threshold stimulation. Signals that activate sensory receptors, generate action potentials, travel through peripheral and central pathways, and produce measurable responses in the nervous system. Responses detectable with electrodes, visible in imaging data, real in every physiological sense, without ever producing conscious awareness of the stimulus that caused them. The body responded. The person did not notice. This is not rare. It is not a laboratory curiosity produced only under artificial experimental conditions. It happens continuously in every waking nervous system. At every moment when the sensory environment contains stimuli that fall just short of the conditions required for conscious detection. The question of what those conditions actually are is one that remains, in certain important respects, genuinely open. One framework describes the threshold in terms of signal strength. The idea that a stimulus must produce a neural response of sufficient amplitude to cross from unconscious processing into awareness. In this view, subthreshold signals are simply too weak, the cascade they initiate too small to recruit the broader network activity that conscious perception seems to require. There is evidence for this. Stimuli near the detection threshold produce cortical responses that are smaller and shorter in duration than stimuli well above it. The neural signature of a barely detected stimulus is measurably different from the signature of one that is comfortably felt. But the relationship between signal strength and conscious detection is not clean. The same stimulus, delivered twice in identical physical conditions, is sometimes detected and sometimes not. The peripheral response, as far as can be measured, is equivalent. What changes is something in the central state of the nervous system at the moment of arrival. Something about the readiness of the receiving circuits, the current level of spontaneous neural activity, the particular configuration of a system that is never truly still. This variability is part of what makes the threshold difficult to pin down. It is not a fixed line. It moves. It moves with arousal, with attention, with the recent history of the circuits involved, with factors that have nothing to do with the stimulus itself, and everything to do with the state of the system that is supposed to receive it. A signal that would have been detected 30 minutes ago in a slightly more alert nervous system may not be detected now, not because it changed, because the system it is arriving at changed. The threshold rose, the signal stayed the same, the signal no longer clears it. There is a theory, influential, debated, not entirely resolved, that attempts to describe what happens at the threshold in terms of neural dynamics rather than signal strength alone. Global workspace theory, developed primarily by Bernard Baars and later extended by Stanislas Dehaene and colleagues, proposes that conscious perception involves the ignition of a widespread network of cortical areas, a sudden, coordinated increase in activity that broadcasts information across the brain in a way that local processing cannot achieve. In this view, subthreshold stimuli are processed locally, in primary sensory cortices, in subcortical structures, but fail to trigger the global ignition that makes information available to the broader system. Conscious experience in this framework is not a property of any single brain region. It is an event, a dynamic state that either happens or does not. The subthreshold signal is processed. It never triggers the event. It's not entirely clear how to think about what this means for the signals your body is generating right now. The receptors in your skin are still active. The signals are traveling. Some are reaching the cortex in diminished form. Whether any of them are triggering anything resembling global ignition, whether any of them are producing even the faint, brief version of that coordinated network activity that barely conscious states sometimes show, is genuinely uncertain. What seems likely, given everything already described, the synaptic depression, the thalamic shift toward burst mode, the decreasing corticothalamic feedback, the rising threshold, is that most of what arrives at the cortex right now meets the cortex in a state that is not prepared to ignite. The signal arrives. The network does not respond. There is something worth noticing in this, something that sits a little differently from the other layers of filtering described so far. The spinal cord filtering, the thalamic gating, the synaptic depression, these are mechanisms. They reduce the signal before it reaches the cortex. They are upstream of experience in a fairly straightforward sense. But what global workspace theory describes is different. It is a failure of ignition in a cortex that has received the signal. The information is there, the local response occurred, and still, conscious experience did not follow. This suggests that the final filter is not a reduction in signal strength, but a change in the cortex's capacity to do something with what it receives. The signal made it through. The cortex could not, or did not, complete the step that would have made it real. The sensation you almost had tonight, the pressure that registered somewhere in your primary somatosensory cortex without ever becoming something you felt, is real in the physiological record. If electrodes were placed at the right locations and the right measurements taken, the response would be there. A measurable deflection in the electrical activity of the cortex, a brief local signature of a signal that arrived and was processed. But you have no access to it. There is no experience associated with it, no memory of it forming, no moment of noticing followed by a moment of forgetting. It did not cross the threshold of ignition. It was processed in the dark, as it were, locally, briefly, without ever becoming available to the rest of the system. The nervous system registered it. You did not. This gap between neural response and conscious experience is one of the oldest and least resolved questions in the study of the mind. It has names: the hard problem of consciousness, the explanatory gap. Researchers approach it from different directions and arrive at different partial answers, and the full answer, if there is one, is not available yet. What is available is the observation itself that your nervous system is right now producing and receiving signals that will never become experience, that the distance between a signal being processed and a signal being felt is not guaranteed to close, that the filtering which began at the surface of your skin continues all the way to the final moment where the brain either does or does not complete the act of perception. Most of what is happening inside you tonight will remain exactly where it is, processed, real, and entirely outside of anything you will ever know about it. The next layer asks a different question. Not what fails to reach experience, but what the brain does with what it expects to receive, the predictions it makes before the signal arrives, and how those predictions shape what, if anything, gets felt at all. A nociceptor does not know it is a nociceptor. It has no access to the category it belongs to, no awareness of the role it plays in the larger system, no sense of the difference between itself and the mechanoreceptor a few micrometers away responding to the same general region of tissue. And yet the two cells are genuinely structurally different, tuned through molecular architecture to respond to different kinds of physical and chemical change, built to generate signals that travel to the brain through different fiber types and arrive at different processing stations, producing, in sufficient quantity and under the right conditions, experiences as distinct as the feel of fabric and the feel of injury. The distinction begins at the membrane. Nociceptors, the sensory neurons whose activation underlies the eventual experience of pain, are not simply low-threshold mechanoreceptors set to a higher sensitivity. They are a distinct cell population with their own receptor proteins, their own ion channel composition, their own thresholds and response properties, and their own particular relationship to the chemical environment of the tissue they innervate. A relationship that changes when the tissue is damaged or inflamed in ways that have no equivalent in the mechanoreceptors sitting nearby in the same region of skin. What stands out most when you look at the molecular profile of a nociceptor is the ion channel TRPV1, transient receptor potential vanilloid 1, a membrane protein that responds to heat above approximately 43 degrees Celsius, to certain acidic conditions, and to capsaicin, the compound responsible for the heat sensation of chili peppers, and the reason these three very different stimuli, thermal, chemical, acidic, produce something that feels phenomenologically like burning, is that they all converge on the same ion channel, the same molecular gateway, producing the same downstream signal regardless of which input opened it. The nociceptor does not distinguish between them, it only knows that the channel opened. When TRPV1 opens, or when any of the other ion channels characteristic of nociceptors reach their activation threshold, sodium and calcium ions flow into the cell along their electrochemical gradients, depolarizing the membrane, generating a receptor potential that, if it reaches sufficient amplitude at the axon hillock, the region of the cell where action potentials are initiated, will trigger the firing of an action potential that travels along the axon toward the spinal cord. This is the moment the signal begins its journey. It is not pain yet. It is an electrical event in a single cell, traveling at a speed determined by the diameter of the axon and the presence or absence of myelin. Fast in the large myelinated delta fibers that carry the sharp, immediate quality of acute pain. Slow in the small unmyelinated C fibers that carry the diffuse, sustained, aching quality that tends to arrive after the first sharp signal has passed. Two signals from the same region of tissue, travelling at different speeds, arriving at the dorsal horn with a temporal separation that is, under the right conditions, large enough to be perceptible. The double pain response, the brief gap between the first sharp sensation and the duller wave that follows it. The nociceptor generated both. It had no experience of either. At the dorsal horn, the nociceptive signal meets the same synaptic architecture described in earlier chapters. The inhibitory interneurons, the competing inputs, the descending modulatory pathways, but nociceptive transmission has features that distinguish it from the processing of ordinary touch, features that become most relevant when the system is operating under conditions of sustained or repeated injury. The primary afferent nociceptor releases, at its central terminal in the dorsal horn, not just glutamate, the fast excitatory neurotransmitter, but also neuropeptides, among them substance P and calcitonin gene-related peptide, molecules that act more slowly than glutamate and over a wider area, diffusing beyond the synapse to influence neighboring neurons and to modulate the excitability of the dorsal horn in ways that outlast the original signal by seconds to minutes. This is one of the mechanisms underlying wind-up, the phenomenon in which repeated nociceptive stimulation at a constant physical intensity produces progressively increasing responses in dorsal horn neurons, a form of short-term plasticity in pain processing that is distinct from the synaptic depression seen in ordinary sensory habituation, moving in the opposite direction, amplifying rather than reducing, preparing the system to respond more strongly to what it has just been repeatedly asked to process. The synapse for pain can learn to be louder. This is, in the most precise sense, the opposite of what is happening in your body right now. Right now, in the absence of tissue damage, in the absence of inflammation, in the absence of sustained nociceptive input of any meaningful intensity, the nociceptors in your skin are largely quiet. Not completely silent, because a baseline level of spontaneous activity exists in most sensory neurons, and because the thermal environment of skin in contact with fabric involves temperature gradients that may produce occasional low-level activation of thermally sensitive nociceptors at the margins of their response range, but quiet in the sense that matters, in the sense that nothing they are generating is producing anything close to what would be required to reach the processing stations where the experience of pain is assembled. The signal, even if it begins, does not go far. What stands out in thinking about nociception from the inside of the neuron rather than from the inside of experience is how much processing intervenes between the molecular event at the membrane and anything that could be called suffering, and how many of the structures involved in that processing are actively working right now to ensure that the signals being generated, whatever small ones exist, do not complete the journey. The periaqueductal grey matter, a region of the midbrain with a well-documented role in the descending modulation of pain, is receiving input from the hypothalamus and the limbic system that reflects the current state of the organism, resting safe, not under threat, and releasing endogenous opioids into descending pathways that reduce the excitability of the dorsal horn neurons that would otherwise relay nociceptive information upward. The body is applying its own analgesia, quietly without instruction, because the conditions indicate that it should. The nociceptors are there, The pathways are intact, the processing stations are functional, and the entire system is, at this moment, actively arranged to prevent them from mattering. There is something almost counterintuitive about this. The recognition that the absence of pain is not simply the absence of nociceptive input, but is in part an active state, maintained by structures that are working to suppress what input does exist, that the painlessness of an ordinary resting body is something the nervous system produces, rather than something that simply occurs when nothing is wrong. The endogenous opioid system, the descending noradrenergic pathways from the locus coeruleus, the serotonergic fibers descending from the raphe nuclei. These are all active participants in the quiet of your body right now, all contributing to an inhibitory tone that holds the dorsal horn below the threshold of meaningful nociceptive transmission, all doing work that produces, from your side of experience, simply nothing. The nociceptor at the membrane does not know this work is being done on its behalf. It only knows the current state of its own ion channels, closed, mostly or briefly open to signals too small to propagate, or opening and closing in the low-level way that all excitable cells cycle through, without generating anything that leaves the cell with enough amplitude to matter. Pain, assembled from the output of these cells and the processing it undergoes across a dozen intervening structures, is perhaps the most vivid evidence that experience is not a direct readout of what is happening in tissue, that the relationship between cellular event and felt sensation is mediated, modulated, filtered, amplified, or suppressed by so many layers of neural processing that the original event bears only a loose relationship to what, if anything, eventually becomes experience. The nociceptor fired. What happened after that was the result of 100 other conditions that had nothing to do with the firing itself. Right now, those conditions are arranged in the direction of silence. The inhibitory tone high, the descending suppression active, the dorsal horn quiet, the thalamus in the early stages of its nightly retreat from sensory engagement, the cortex withdrawing from the processing of peripheral input. The cells at the surface of your body are doing what cells do, maintaining membrane potentials, cycling through baseline activity, responding to the ordinary thermal and mechanical conditions of a body at rest. None of it is becoming pain, none of it is becoming much of anything. And the system that ensures this is not passive, not the mere absence of cause. It is an active, distributed, continuously maintained arrangement of circuits whose combined output is the simple, unremarkable, deeply ordinary experience of a body that is, for now, comfortable enough to let everything else go quiet. The signal hasn't arrived yet, and the brain is already preparing a response to it. This is not a metaphor for anticipation, not a poetic description of readiness. It is a literal account of what the nervous system does continuously in real time, using the history of what has arrived before to generate predictions about what is about to arrive next, and then using those predictions to pre-shape the circuits that will receive the incoming signal when it does come, so that the actual sensory input, when it finally arrives, is not processed from scratch but evaluated against something the brain has already quietly constructed in advance. The term used in contemporary neuroscience is predictive coding. The implications of it take a while to settle. The framework developed most prominently by Carl Friston, though with roots in earlier work by Helmholtz and later by Rao and Ballard, proposes that the brain is not primarily a sensory receiver but a prediction machine, an organ whose fundamental operation involves generating internal models of the world, comparing incoming sensory signals against those models, and updating the models when the signals carry information the prediction did not anticipate. What gets processed most completely in this view is not the signal itself, but the difference between the signal and the prediction error. What the brain predicted correctly, what arrived exactly as the model said it would, requires very little processing. It confirms what was already known. It carries no new information. It is, in a precise computational sense, redundant, and redundant signals the brain tends to suppress. You've been in this position for a while now. The pressure of the surface beneath you has been consistent. The temperature at the skin has reached equilibrium. The acoustic environment, whatever it contains, has been stable long enough that its pattern has been internalized. The model your brain maintains of your immediate sensory environment has had time to become accurate. Accurate enough that most of what arrives from the periphery now matches what the model predicted would arrive. The prediction error is close to zero, and when prediction error approaches zero, the ascending sensory signal, the one travelling from the periphery toward the cortex, is dampened. Not by a separate suppression system, but by the structure of the predictive coding architecture itself, which uses descending signals from higher cortical areas to cancel out the predicted components of ascending sensory input before those components reach full cortical processing. The signal arrives. It matches the forecast. The cortex, in a sense, already knew. And what it already knew, it does not need to process again. This is, and it takes a moment to find the edges of this idea, a different kind of filtering from everything described so far. The spinal cord filtering was about competition between signals. The thalamic gating was about arousal state and relay mode. The synaptic depression was about the history of individual junctions. But predictive coding describes filtering that is based on knowledge, on the brain's accumulated model of the sensory world, applied in real time to reduce the processing cost of confirming what was already expected. The brain is not simply ignoring the familiar, it is actively predicting it, and using the prediction to preemptively cancel the signal, so that only what is genuinely surprising requires a full response. This is, in a strange way, more intimate than the other mechanisms. It means that what you feel is shaped not just by what is happening at your skin, but by what your brain has learned to expect. That perception is partly a readout of the model, not just the signal, though what reaches experience is in some meaningful sense a construction rather than a reception. The history of predictive coding as a framework is interesting partly because it converges on conclusions that seem at first almost too strange to be biological. The idea that the brain hallucinates its sensory world and then corrects the hallucination using incoming data. That ordinary perception is a controlled form of prediction rather than a faithful representation of external reality. Hermann von Helmholtz, in the 19th century, described perception as unconscious inference, the brain drawing conclusions about the world from incomplete sensory data, using prior knowledge to fill in what the senses did not fully provide. He was working without the tools to describe the mechanism, but the inference he drew about perception has held up in ways that more precisely instrumented researchers have since confirmed. The brain infers, it constructs, it predicts, and when the prediction is good enough, the sensory signal that arrives to confirm it is suppressed before it can do much of anything at all. There are neurons in the visual cortex, and by extension in other sensory cortices, whose firing rate decreases when a stimulus is presented that matches what the surrounding context predicts. They respond vigorously to unexpected inputs and fall quiet when the input is expected. These neurons are not faulty. Their silence is the signal. The silence means, this is what was predicted, nothing new here, no update required. The neurons that fire most are the ones reporting surprise. The ones reporting confirmation go quiet. Right now, in the somatosensory cortex processing the input from your skin, the neurons that would respond most vigorously to your current sensory environment have already been through several cycles of prediction and confirmation. The model of the pressure, the temperature, the texture, it was built in the first minutes of contact and has been refined and confirmed repeatedly since. The prediction is good. The neurons reporting confirmation are quiet. What fires, if anything fires with any completeness right now, is the occasional deviation, a small shift in pressure as you breathe, a brief change in the temperature gradient at an exposed area of skin, something the model did not quite predict, something small enough to produce only a faint prediction error signal, which is processed briefly and then incorporated into the model, which then predicts it correctly the next time, which then suppresses it. The environment is being absorbed into the model, and once it is absorbed, it effectively disappears. Sleep does something specific to this process, something that is still being worked out in the literature, not entirely resolved, which is part of what makes it worth pausing on. During sleep, the brain's predictive models are believed to undergo consolidation and updating. The accumulated prediction errors of the day are processed, the models are revised, the parameters that determine what the brain expects from its sensory environment are adjusted based on what the day's experience contained. This is one of the proposed functions of sleep that has accumulated the most supporting evidence, that sleep is when the model gets updated. When the predictions for tomorrow are built from the experience of today, the brain in the process of falling asleep is beginning to release its grip on the sensory predictions it has been running all day, the models of the immediate environment, the ongoing calibration of sensory expectations, and shifting into a mode in which prediction, in the usual sense, is temporarily suspended. What replaces it during deep sleep is still not fully understood. What is clear is that the predictive suppression of sensory signals, which has been running continuously since you settled here, is about to change character. The descending signals that cancel predicted sensory input will shift. The cortical predictions will loosen. The model will begin, in its own quiet way, to dissolve. And in the space where the model was, where the prediction had been holding the sensory world at arm's length, something else will begin. Not silence exactly, but a different quality of internal activity, one that has less to do with the surface of the body and more to do with whatever the brain does when it is no longer in the business of confirming what it already knows. Something is touching you right now that you cannot feel. You know this in a way. Not because you checked, not because you reasoned through it, just because you've been in a body long enough to know that sustained contact fades, that the thing pressing against your back or your side or the back of your hand has, at some point in the last hour, simply stopped being present in any way that registers. It didn't go anywhere, you just stopped feeling it. The receptors responsible for registering sustained gentle pressure are called slowly adapting mechanoreceptors. There are two main types. Merkel discs sitting close to the skin surface respond to static pressure and fine spatial detail and maintain their firing for as long as the stimulus is present. Ruffini endings deeper in the dermis respond to skin stretch and sustained pressure, particularly at the edges of contact where the skin deforms most. Both fire continuously under sustained touch. Neither produces the experience of sustained touch for very long. This is the part that takes a moment to sit with. The cells are doing their job. The signal is being generated. The fiber is carrying it toward the spinal cord with the same fidelity it always carries things. And yet the sensation disappears. The disappearance happens not at the receptor but further along, at the synapses in the spinal cord where the signal first meets the central nervous system, and at the thalamic relay, and at the cortex that has already been through this exact signal enough times tonight to have substantially reduced its response to it. What the receptor reports and what the cortex eventually receives are not the same thing. They stopped being the same thing some time ago. There is a distinction worth making here between two kinds of adaptation that are easy to conflate. Rapidly adapting receptors, Meissner's corpuscles and Pacinian corpuscles, physically stop responding to sustained stimuli. Their mechanical structure is designed to respond to change, to motion, to onset and offset. Under sustained constant pressure, their firing rate drops to near zero within milliseconds to seconds. The receptor itself adapts. The signal stops being generated. Slowly adapting receptors don't do this. They keep firing. The adaptation that produces the disappearance of sustained touch is not happening at the receptor. It is happening centrally, in the processing chain, at the synapses, in the circuits that decide what to do with a continuous, unchanged, low-priority signal. The receptor is still speaking. The brain has stopped listening. You felt the reversal of this. Shifting position makes the contact suddenly present again, not because the pressure increased dramatically, but because the distribution changed, because a set of receptors that had been firing continuously at a stable rate suddenly had to adjust to a new pattern, and that adjustment produced prediction errors in the cortical model that had been calmly forecasting exactly what was arriving. The unexpected is felt. The expected disappears. This is what returning to an environment after an absence can do. Make a smell vivid again that had been absent from awareness for hours. Make a sound present that had become invisible. Make the temperature of a room suddenly register after it had been gone. Nothing changed in the environment. The model was reset. The prediction errors returned. There is a specific study worth describing here, not because it changed everything, but because it demonstrates something cleanly. Researchers applied sustained, gentle pressure to participants' fingertips, pressure below the pain threshold, steady, constant, and asked them to report when they could feel it. Within a few seconds to a minute, most participants reported that the sensation had faded or disappeared entirely. Electrode recordings from sensory cortex showed that the cortical response to the sustained pressure had decreased substantially from its initial amplitude. Then researchers introduced a tiny, brief change in the pressure, not removal, just a slight variation. The sensation returned immediately. The cortical response recovered. The receptor had been firing the whole time. The cortex had simply been declining to represent it fully. One small change was enough to re-engage the full processing that the unchanging signal had lost. The contact your body is in right now has not changed for long enough that this process has run its full course. The slowly adapting receptors are still firing. The fibers are still carrying the signal. The spinal cord has seen this pattern enough times in the last hour that its response has shortened. The thalamic relay has adjusted. The cortical prediction is accurate to the point of near total suppression. What you feel, if anything, at the surface of your body right now is the residue, the occasional small deviation from the expected pattern, the slight shift in pressure as your breathing moves your chest, the tiny temperature fluctuation at the edge of a covered and uncovered region of skin. The rest has been absorbed into the model and suppressed. This is not a failure of attention. It is not inattentiveness or distraction. It is the nervous system operating as it should, conserving the processing resources that sustained, stable, non-threatening contact does not justify consuming, maintaining just enough representation of the body's contact with its environment to detect genuine changes and releasing everything else from the cost of full experience. The touch that disappears is still there. It never stopped being there. It became at some point that had no clear beginning simply not worth the energy of feeling. You're in contact with something right now that you are not feeling. This is ordinary. This is the body working correctly. The surface that holds you has been present long enough to be known, expected, incorporated, present enough to be safe, stable enough to be ignored, continuous enough to be released. What remains at the edges of your sensory awareness right now is only what is still changing. Small, slow, barely enough to register. And those edges are getting quieter. The next layer of this is not about the skin at all. It is about what happens when the filtering system itself begins to be filtered, when the resources that maintain the suppression of sensation start to thin, and the boundary between what is suppressed and what is felt begins, in certain specific conditions, to move in an unexpected direction. The filtering system has limits. This is not a flaw in the design. It is a necessary property of any system that uses active suppression rather than passive absence. Active suppression requires resources. It requires the continuous provision of inhibitory tone, the ongoing activity of descending modulatory pathways, the sustained operation of circuits that consume energy to produce silence. When the resources that maintain the suppression begin to thin, the suppression itself begins to thin with them. What was background can become foreground. The clearest demonstration of this is familiar to most people, though rarely identified as a sensory phenomenon. Extreme fatigue, not the ordinary tiredness of a long day, but the particular quality of exhaustion that arrives after sustained sleep deprivation or prolonged physical stress, produces, among other things, a change in sensory experience that most people describe as a kind of rawness. Sounds become more intrusive. Light becomes less comfortable. The feel of clothing against skin, ordinarily absent from awareness, returns with an insistence that is difficult to locate as either pain or touch. The signal from the periphery did not change. The suppression that had been holding it at bay weakened. The mechanism involves in part the same descending modulatory pathways described throughout this chapter series. The noradrenergic fibers descending from the locus coeruleus, the serotonergic fibers from the Rafe nuclei, the endogenous opioid release from the periaqueductal gray, these systems are not inexhaustible. They operate within the constraints of the neurotransmitter synthesis and release cycles that govern all chemical signaling in the nervous system, and under conditions of prolonged activation, their output can decrease. The locus coeruleus, in particular, shows reduced firing rates after sustained periods without sleep. Its output to the spinal cord and thalamus, output that contributes to the maintenance of inhibitory tone in sensory processing, decreases. The tone drops. The threshold that had been holding routine signals below conscious awareness moves downward. Signals that had been suppressed now clear it. This is, in a specific biological sense, the filter reversing, not dramatically, not in a way that produces acute pain from ordinary contact under most circumstances, but in a way that changes the quality of sensory experience, makes it denser, more present, less easily dismissed. The room feels louder. The body feels more immediate. The background that had been reliably silent begins to produce a low, continuous presence that was not there before. I tend to think this is one of the more underappreciated consequences of sleep deprivation, not the cognitive effects which are well documented, but this particular sensory quality, the way the world becomes slightly too much, the way ordinary stimulation starts to cost something that it did not cost before. The nervous system is running the suppression on diminished reserves. There is a condition called allodynia, mentioned briefly in the chapter on chronic noise, that represents a more sustained version of this reversal. In allodynia, stimuli that are not painful under normal circumstances produce pain. Light touch, clothing contact, air movement across the skin. The sensory threshold has shifted far enough that the ordinary sensory environment becomes a source of discomfort that healthy sensory processing would have filtered without effort. Allodynia occurs in several chronic pain conditions, in certain neuropathies, in fibromyalgia, conditions that share, among other features, evidence of reduced inhibitory tone in the central sensory processing pathway. The dorsal horn interneurons that would normally suppress the upward transmission of touch signals are less active. The descending modulation from the brainstem is less robust. The thalamic gating is altered. The result is a nervous system that cannot perform the suppression it is supposed to perform, and a sensory world that reflects that inability directly in experience. The ordinary case, the case of a healthy nervous system approaching sleep, involves something gentler but structurally related. As wakefulness decreases and the brainstem's arousal systems reduce their output, there is a brief window during the transition to sleep in which certain sensory thresholds shift before the sleep-specific suppression mechanisms fully engage. The thalamic gating has not yet reached the depth it will achieve in light sleep. The cortex is partially disengaged but not yet fully isolated from thalamic input. The descending noradrenergic tone has begun to decrease. For a short time, certain signals that were suppressed during alert wakefulness and will be more strongly suppressed during established sleep exist in a window where neither suppression system is fully operational. This is part of why the transition to sleep can, in some people, involve a brief period of increased sensory awareness, a moment of noticing the room more clearly or feeling the body more immediately before the sleep-specific mechanisms deepen and the sensory world recedes again. There is also something worth noting about illness, about the particular sensory quality of a fever or a systemic infection that most people recognize but rarely examine closely. During significant immune activation, pro-inflammatory cytokines, signaling molecules released by immune cells, cross into the central nervous system and modulate the activity of neurons in ways that include reducing the efficacy of inhibitory signaling in sensory processing circuits. The same descending inhibitory systems that maintain sensory suppression under normal conditions become less effective in the presence of high cytokine concentrations. This is why illness makes the body feel louder, why the weight of a blanket becomes noticeable, why light seems too bright and sound seems too present. The immune system, doing its necessary work, has as a side effect temporarily altered the sensory filtering capacity of the nervous system. The body is not generating more sensation. It is suppressing less of what it generates. Tonight, the conditions are different. The descending modulatory pathways are not depleted. The inhibitory tone in the dorsal horn is intact. The immune system is not releasing cytokines in concentrations that alter central sensory processing. The thalamic gating is deepening rather than weakening. The filter is not reversing. It is strengthening, moving in the direction it moves every night in a resting, healthy body approaching sleep. The threshold is rising. What was faintly present at the edges of sensory awareness 20 minutes ago is already less present now. The signals that were just barely registering are falling below a threshold that has continued to move upward as the sleep transition proceeds. The filter is not at risk tonight. It is doing exactly what it was built to do, maintaining the suppression through the transition, handing off from waking mechanisms to sleep mechanisms without creating the gap that would allow the ordinary sensory environment to reassert itself. The body you are in right now is not raw, it is not sensitized, it is not running its suppression systems on diminished reserves. It is a body at the far end of a long filtering process, in which signals have been reduced at the receptor, at the synapse, at the spinal cord, at the thalamus, at the cortex, and are now being held below the threshold of experience by a system that has everything it needs to continue doing so through the night. The world at the surface of your skin is still arriving. The receptors are still reporting. The fibers are still carrying their signals inward. But the system receiving them is, at every level, less and less interested, and the interest will keep falling, slowly, in the direction it's been falling since you settled here. until the sensory world outside the body becomes something the brain is no longer in the business of representing at all. What remains when that happens, what the brain attends to when it has released the surface of the body entirely, is the subject of what comes next. The body is monitoring itself right now, not you, not any part of you that has a name or a voice or a preference about what happens next. The body, the distributed network of sensors and feedback loops and signaling pathways that runs continuously beneath anything that could be called awareness, is checking its own conditions with a thoroughness that no external observation could match. Blood pressure at multiple points in the arterial tree, oxygen saturation in the tissues, pH in the cerebrospinal fluid, stretch in the walls of the stomach and intestines, tension in tendons and joint capsules, temperature at the body's core versus its surface, the concentration of glucose in the blood passing through the hypothalamus, all of it right now being measured, almost none of it reaching you. The sensors involved in this internal monitoring are as real and as structurally sophisticated as the mechanoreceptors in the skin, but they are, for the most part, organized around a principle that is almost the opposite of the principle governing cutaneous sensation. Skin receptors are built to report to consciousness. Their signals travel pathways designed to produce experience, to generate the felt awareness of touch, temperature, pressure, pain. The architecture of the cutaneous sensory system is oriented ultimately toward making the surface of the body present to the mind that inhabits it. Introceptive receptors, the sensors monitoring the body's internal state, are built mostly to report to other body systems rather than to consciousness. The baroreceptors in the carotid sinus and aortic arch send their signals primarily to the brainstem cardiovascular control centers. The chemoreceptors monitoring arterial oxygen and carbon dioxide report to the respiratory control centers in the medulla. The stretch receptors in the gut communicate with the enteric nervous system, with the vagus nerve, with autonomic ganglia, a conversation that happens almost entirely without cortical involvement. The organs do not particularly want the brain to know what they are doing. They want to be left alone to manage it. This is a hierarchy, not a rigid one, not one without exceptions, but a consistent organizational principle in which internal signals are routed, by default, away from conscious processing and toward the regulatory systems that can act on them directly and efficiently, without the delay that cortical involvement would introduce. The baroreceptors detect a drop in blood pressure, and the signal reaches the brainstem in milliseconds, producing a compensatory increase in heart rate and vascular tone before anything resembling awareness has had time to form. The chemoreceptors in the medulla detect a rise in carbon dioxide, and the respiratory rate increases, again, before you know anything about it. The body corrected itself. You were not consulted. There are conditions under which internal signals do reach consciousness, and the conditions are informative about the hierarchy's logic. Pain from visceral organs, the gut, the kidneys, the heart, reaches awareness when the stimulus is severe enough, or the tissue damaged enough that the normal routing to subcortical regulatory centers is insufficient, when the situation has moved beyond what autonomic management can address, when the brain needs to know in order to produce a behavioral response. The pain is not proportional to the severity of the internal event in the way cutaneous pain tends to be proportional to the severity of skin injury. Visceral pain is diffuse, poorly localized, often referred to body surface areas that share spinal cord segments with the affected organ. It is not designed for precision, it is designed for urgency, to cross the threshold into awareness when something internal has moved beyond the range of quiet self-regulation. The signal that reaches consciousness from inside the body is, in most cases, an escalation, a request that has been refused at every lower level and is now arriving loudly at the only level with the behavioral resources to respond. Most internal signals never become that request. The interoceptive pathway that has received the most research attention in recent years is the one travelling through the vagus nerve. The 10th cranial nerve, a long branching structure that carries signals from the heart, lungs, liver, stomach, intestines, and other visceral organs toward the brainstem, and through the brainstem toward cortical regions including the insula. The insula is the cortical area most consistently implicated in the conscious representation of body states, in what researchers call interoceptive awareness, the capacity to perceive internal signals. It receives input via the thalamus from the vagal pathway and from other interoceptive routes, and it is active during the conscious experience of heartbeat, of hunger, of nausea, of the particular felt quality of emotional states that have a somatic component. But the insula receives only a fraction of what the vagus carries. The vast majority of vagal afferent signals, the signals travelling from body to brain rather than brain to body, which constitute roughly 80% of vagal fibres, are processed subcortically, routed to the nucleus tractus solitarius in the brainstem, and from there to the regulatory centres that act on them without ever producing an experience the insula would recognise. 80% of what the vagus reports never reaches anything like awareness. What does reach awareness, via the insula and related cortical regions, is shaped by more than just signal strength. It is shaped by attention directed inward, by the learned tendency to notice or not notice internal states, by the current overall level of interoceptive sensitivity, which varies between individuals and within individuals across different states. Right now, directed inward, there are signals available that are not normally present to awareness. A faint sense of the heartbeat if you attend to it, a background of abdominal sensation that would not ordinarily register, a quality of internal warmth or the weight that is always there but rarely noticed. It's not entirely clear what determines exactly how much of this reaches awareness under ordinary conditions versus which conditions allow more of it through. The research on interoceptive sensitivity suggests that it involves both the strength of the ascending interoceptive signals and the degree to which higher cortical areas, particularly the prefrontal cortex, are directing attention toward or away from internal versus external sources of information. As the prefrontal cortex begins its nightly disengagement, as external attention decreases, there is sometimes a brief window in which internal signals become more noticeable, not because they increased, but because the attentional balance shifted. The body running itself tonight is doing so with the same thoroughness it maintains every night, every hour, every moment of every day. The baroreceptors cycling, the chemoreceptors monitoring, the stretch receptors in the gut reporting, the vagal afferents carrying their continuous update of the interior to a brainstem that processes it without asking for permission or confirmation from anywhere above. The cardiovascular system is making small adjustments to maintain blood pressure, as your position and the distribution of your blood volume change slightly with each breath. The respiratory centers are maintaining a rhythm that will, as sleep deepens, slow and regularize without any conscious contribution. The gut is doing whatever the gut does at this hour. Motility patterns that shift between waking and sleep states, secretory activity that follows its own circadian rhythm, a continuous dialogue between the enteric nervous system and the vagal pathway that has almost nothing to do with what you are experiencing or not experiencing right now. The body does not need you for any of this. It has been doing this since before there was a you in any meaningful sense, since the early weeks of development, when the autonomic nervous system was already establishing the feedback loops that would maintain the organism long before the cortex existed to supervise them. What is worth noticing, if anything, is simply how much is happening, not dramatically, not in a way that demands attention or generates experience, just continuously, quietly, with the precise and unhurried competence of systems that have been doing this for longer than memory, longer than thought, longer than any of the structures capable of recognizing what they are doing have existed. The hierarchy places almost all of it below the level of awareness by design, not to hide it, not because it is unimportant, but because the awareness that would receive it has better uses, and because the systems managing it do not need awareness to do their work well. They need only the conditions they currently have: a body at rest, a chemistry within its normal ranges, a nervous system that is, at this hour, releasing its grip on the external world and leaving the interior to the quiet, competent management of systems that were never waiting for permission to begin. The next chapter will take us to the edges of the sensory field, to the signals that exist at the margins of what the nervous system can detect that are real but unclassifiable, that the brain sometimes resolves and sometimes simply leaves undefined. There is a region at the margin of what any sensory system can detect where the usual categories stop applying cleanly. Not pain, not touch, not temperature in any precise sense. Something that arrives at the surface of awareness, if it arrives at all, without the label that more central, more intense stimulation tends to carry with it. The nervous system encountering a signal at this margin does something that is neither full processing nor full suppression. It makes a partial attempt at classification, and then, finding no category that fits well enough, sometimes leaves the signal in a kind of holding state that never resolves into a named experience. This region exists. It has been measured in various ways in various sensory modalities, and almost everyone has been in it without knowing it had a name. The absolute threshold, the minimum stimulus intensity required to produce a conscious sensory experience under ideal detection conditions, is not a fixed property of the sensory system, but a statistical description of its behavior, defined classically as the intensity at which a stimulus is detected 50% of the time across repeated presentations, which means that by definition, half of the stimuli at this intensity are not detected, and that below this intensity, detection becomes progressively less likely without ever becoming impossible, and that the region between 0 probability of detection and 50% probability is a real continuous stretch of physical stimulus space in which signals are generating neural responses without reliably generating experience. This gradient is what the edges of the sensory field actually are, not a line, a probability distribution shallow at the low end steepening toward the threshold, populated throughout by signals that exist in the nervous system, but whose relationship to experience is genuinely uncertain. Signal detection theory, developed in the 1950s and 1960s in the context of radar operator performance, and later applied extensively to sensory psychology, provides a framework for separating two components that the classical threshold concept conflated: the sensitivity of the sensory system to a signal and the criterion the observer uses to decide whether to report detecting it. What this framework revealed was that the sensory boundary is not purely a property of the stimulus, or even of the sensory system, but involves a decision process, a threshold of report that is set, implicitly and continuously based on factors including the expected probability of a signal being present, the costs and benefits of false alarms versus misses, and the current state of the nervous system that is doing the detecting. The same physical signal at the same intensity can be detected or not detected depending on where the decision criterion sits. And the decision criterion moves. It moves with arousal, with attention, with expectation, and it moves with the state the nervous system is currently in, which is a state of decreasing sensitivity to external input, increasing threshold, decreasing willingness to commit to the report of a sensation that is not clearly present and insistent. The criterion tonight is set conservatively. What would have been reported as a faint but definite sensation an hour ago might now fail to cross the criterion for report, not because the sensory system has become less sensitive in a peripheral sense, but because the decision-making layer that sits above raw sensory processing has raised the bar for what counts as real enough to acknowledge. This is part of why the sensory world feels thinner right now. The world has not changed. The decision about what counts has. There are sensory experiences that resist classification not because they are weak, but because they genuinely occupy the space between the named categories the nervous system uses to organize sensation. Thermal sensations near the neutral temperature of the skin, neither warm nor cool, not quite neutral either. A kind of thermal ambiguity that the thermoreceptors register, but that the cortex has difficulty placing. Pressure sensations at the boundary between touch and a deeper proprioceptive quality. Not quite the surface, not quite the joint, somewhere in the tissue that doesn't have a clear felt address. Sensations arising from the fascia, the connective tissue that wraps muscles and organs, which has its own innovation. Mechanoreceptors and nociceptors embedded in tissue that most people have no felt map of, producing signals that arrive, when they arrive at all, without the localization and quality that skin sensation carries. These are at the edges, not weak versions of known sensations, genuinely marginal in their category. The fascia deserves a moment here because it is one of the more interesting territories at the edge of the sensory field. Extensively innervated, physically continuous throughout the body, capable of generating signals in response to mechanical deformation, to sustained tension, to the slow creep of tissue under prolonged loading, and yet almost entirely absent from the felt map that most people have of their own bodies. Fascia contains both myelinated and unmyelinated sensory fibers. Researchers have found Golgi tendon organ-like receptors, Ruffini endings, Pacinian corpuscles, and free nerve endings distributed through fascial tissue, suggesting a sensory capacity that is structurally present and functionally active. There is growing evidence that facial mechanoreceptors contribute to proprioception, to the body's sense of its own position and movement, in ways that supplement the better-known contributions of muscle spindles and joint receptors, and yet the felt contribution of fascial sensation to ordinary body awareness is, for most people in most conditions, essentially zero. The signals are there, the pathway exists. What stands out in looking at this is how consistently the cortex fails to build a felt representation from what is, structurally, sufficient input to support one. Whether this is a learned absence, a map that was never built because the signals were never attended to, or something more fundamental about the way facial sensory information is processed before it reaches the cortex, is not entirely resolved. Right now, in the sustained stillness of your current position, there are mechanical signals arising from the slow creep of tissue under the loading of your body's weight, from the fascia deforming gradually, from the connective tissue of joint capsules under sustained mild tension, from the tendons of muscles that are neither actively contracted nor entirely slack but somewhere in the resting state between the two. These signals are arriving at the spinal cord. Some are reaching the thalamus. A fraction may be reaching the cortex in some form. None of them are likely to become anything you could point to or describe. They are at the edges, real as any other biological signal. Measurable if the right electrodes were placed in the right locations, completely absent from the felt map of a body that's been still long enough for the unusual to become ordinary and the marginal to become invisible. The sensory field has edges, and those edges are not fixed, and tonight they have moved inward considerably from where they were this afternoon, and the territory that now lies outside of felt experience is larger than it was. more of the body's surface, more of its interior, more of the signals it generates in the ordinary course of being a living tissue under mild mechanical load in a stable thermal environment. The brain is representing less of the body than it usually does, not because the body is generating less, because the threshold has risen, the criterion has shifted, and the region of signal space that crosses into felt experience has contracted toward only what is most insistent, most changed, most different from what was already known. Almost nothing qualifies right now, and the territory of the unrepresented, the edges and margins and sub-threshold signals and unclassified sensations that are running continuously without ever becoming experience, is as close to the whole of the sensory field as it ever gets during waking life. The last chapter will not move outward from here. It will move with what is already happening, with the particular sequence in which the nervous system releases the sensory world, layer by layer, in the direction it's been moving all along. The sequence has a shape. It does not announce itself. There is no moment of transition that declares itself as the boundary between waking sensory experience and whatever comes after. But the sequence is real, measurable in the electrical activity of the brain, traceable in the behavior of the thalamic relay cells, visible in the progressive withdrawal of cortical responsiveness from the body's surface inward, and it follows a pattern that is consistent enough across individuals and across nights to be described, if not perfectly predicted. Some things quiet first, some things quiet later. The order is not arbitrary. The first to go in most people is the detailed spatial resolution of touch. The primary somatosensory cortex maintains, during wakefulness, a fine-grained representation of the body's surface, the capacity to distinguish 2 points of contact a few millimeters apart on the fingertip, to localize a touch to within a small area of skin, to perceive the texture and edge quality of what is in contact with the body. This fine resolution depends on sustained, high-fidelity thalamocortical transmission and on the active engagement of the cortical circuits that process spatial detail. As the thalamic relay cells shift toward a burst mode, this fidelity decreases. The spatial map blurs. Not all at once, the gross representation of body position, the sense of where the limbs are in space, persists considerably longer, but the fine detail, the capacity to feel texture and edge and precise location begins to fade first. You start to notice this some sense as a kind of softening at the edges of bodily awareness, the surface of the body becoming less specific, less detailed, less localized. What persists longer is proprioception. The muscle spindles and Golgi tendon organs that signal limb position and muscle tension are fed by different thalamic pathways than cutaneous sensation, and these pathways maintain their function deeper into the sleep transition. The body's sense of its own configuration, where the arms are, the angle of the joints, the distribution of weight, remains available longer, the surface detail of what the skin is touching. This makes a kind of evolutionary sense. A body that lost its sense of position before its sense of texture would be a body that could not respond to postural threats during the vulnerable window of sleep onset. The sequence that keeps position available longer than texture is a sequence that maintains, for as long as possible, the information most relevant to survival in the transition. Texture goes first, position stays. Temperature awareness follows a different timetable. The thermal sensory pathway, travelling partly through different spinal cord laminae than the touch pathway, processed in the thalamus by partially distinct nuclei, represented in the cortex with overlap with but separation from the somatosensory touch map, maintains function into light sleep in ways that cutaneous touch does not fully match. This is why cold can wake a sleeping person when touch cannot, why a significant drop in environmental temperature produces arousal from light sleep when an equivalent change in tactile stimulation would not. The thermal pathway has a different threshold for suppression, a different relationship to the arousal systems that maintain alertness, a different role in the survival calculus that shape the sleep architecture the nervous system currently uses. Temperature quiets, but later. It's easy to forget that the warmth you feel, if you feel anything right now, is one of the last things still arriving. Pain, or rather the capacity to respond to nociceptive input, persists longest of all the cutaneous modalities, not because the peri-aqueductal grey matter and its descending opioid pathways stop functioning during sleep, but because the threshold for arousal from nociceptive stimulation remains lower than the threshold for arousal from touch or thermal stimulation throughout the sleep cycle. A sleeping body can be moved, can have its temperature change moderately, can be touched repeatedly, and remain asleep. A sleeping body experiencing significant nociceptive input tends to wake, or to shift toward a lighter sleep stage, as a reflex that operates below the level of the cortex and does not require conscious processing to initiate. The nociceptive pathway is the last guard. It quiets last because it is the one most worth keeping, uninterrupted, the cardiovascular uninterrupted. The cardiovascular baroreceptors do not pause. The respiratory chemoreceptors do not reduce their vigilance. The hypothalamic sensors tracking glucose and temperature and osmolarity maintain their continuous sampling through every sleep stage, through the deepest slow-wave sleep, through REM, through the lightest transitional periods at the edges of each cycle. These systems do not have an off state. They were operating before the cortex existed to receive their reports, and they continue operating when the cortex is as close to offline as it ever gets in a living body. The deepest quiet of sleep is not the quiet of a system that has stopped. It is the quiet of a system that has handed off, transferred the representation of the body's state from the cortical structures that generate experience to the subcortical structures that generate regulation, from the systems that produce feeling to the systems that maintain function. Right now you are somewhere in this handoff. The fine spatial detail of cutaneous sensation has already softened. The texture of what holds you has been gone from awareness for some time. The edges of the body, the precise locations where skin meets fabric, where one region of temperature meets another, have blurred into something less specific, less addressable. What may still be present faintly is some version of position, the broad configuration of the body, the general sense of where weight rests, the slow proprioceptive background that the muscle spindles maintain without effort or instruction. And warmth, perhaps. Some residual thermal awareness at the boundary between covered and uncovered skin, where the gradient still exists and the thermoreceptors still have something to report. These will go too, not now, perhaps, but in the direction everything has been going. The sequence the nervous system follows in releasing the sensory world is not a countdown. It does not move towards zero. It moves toward a different kind of representation, one that is less about what the body is touching and more about what the body is doing, less about the surface and more about the interior, less about the felt presence of a body in a room, and more about the sustained subcortical maintenance of a body that continues its work without needing to be felt at all. Sleep is not the absence of biology. It is biology operating without an audience. The thalamus will continue its rhythmic activity through the night, generating the slow oscillations that coordinate the consolidation of the day's synaptic changes. The hippocampus will replay compressed sequences of the day's experience, forwarding information to the cortex for long-term storage. The glymphatic system, the brain's waste clearance network, most active during slow-wave sleep, will move cerebrospinal fluid through the interstitial space of the brain, clearing the metabolic byproducts that accumulate during waking neural activity. The body has work to do tonight that has nothing to do with sensation. It has been waiting, in a sense, for the sensory processing to quiet enough to begin. What quiets last is not a sensation but an orientation. The residual sense of being a body located somewhere, held by something surrounded by a particular quality of air and temperature and sound. This orientation persists lightly into early sleep, a loose background awareness that is less experience than posture, less sensation than position, less feeling than the faint biological record of a body that knows, without knowing it knows, where it is. And then this too softens, not into nothing, into the different activity of a nervous system that has completed its release of the external world and turned, fully and without remainder, toward the interior work that only happens here, in this particular quiet, at this particular depth, in the hours that return the body to whatever it needs to be before morning asks it to begin again. You started this video with a body full of signals that were already being filtered before you noticed them. You've travelled through every layer of that filtering, from the receptor at the skin surface to the synaptic junction in the spinal cord, from the thalamic relay to the cortical map, from the predictive model to the decision criterion, from the ancient habituation of a flatworm to the global workspace of a brain that either ignites or doesn't. All of it, running quietly inside you every moment of every day, without asking for anything. Tonight it asked for even less. It is asking for nothing now. If this video found you at the right moment, if the biology carried you somewhere restful, if the slow movement through the layers of the sensory system gave your mind something to follow until it stopped following, then it did what it was made to do. The sleepy biologist exists because of the people who return to it, who share it with someone who needs a quiet hour, who become members and leave kind words and press the like button, not as a transaction, but as a small signal that this kind of content is worth continuing. If you are still awake enough to hear this, thank you for being here. A membership, a super thanks, a comment about where you listened from tonight, or simply the act of subscribing so the next video finds you. All of it keeps this going. Sleep well, the biology will continue without you.